Diversity of Life Investigation 3.1a, the Elodea plant. Hi everybody, it's Mr. Kennard. As last lesson, Miss Hubbard brought you up to speed on the use of the microscope. The microscope allows us to observe things that are too small to see with our naked eye. Today, we're going to be using microscopes to look at living material to see what we can learn. We're going to start by looking at the leaves of a common aquatic plant called an Elodea, or Elodea, depends on how you pronounce it. So that's what we're going to be using as our first inclination to see what plant material is made up of. Here's an illustration of what an Elodea plant looks like. You see them sometimes in aquariums, etc., but they're just a simple, very aquatic plant, which means they grow in water, and people put them in their aquariums for fish to eat off of and everything. They also provide us with a great opportunity to look at what microscopic structures make up organisms such as Elodea, and that turns us into our focus question. The focus question for this investigation is, what microscopic structures make up organisms such as Elodea? For this part of the investigation, you'll need to download the notebook sheet 11, which is titled Looking at Elodea. The Elodea lab and the plant we're going to be looking at has two sides. It will have, yes, the top or the bottom, and each one of those is very important as we move through. We're going to be making something called a wet mount, and that will help us to see the various pieces and materials and structures microscopically of what makes up this Elodea plant. With the Elodea plant, as well as many other types of things we'll look under with a microscope, we're going to make what's called a wet mount. It is a lab technique that we use with the slides for the microscope to produce an environment that is easy to see things moving around in water. Here is the video that's going to go along with that.
Wet mount slides are useful when you want to look at living organisms with a microscope. Begin by placing a drop of the culture medium on the slide. Touch the edge of the cover slip to the edge of the drop, then gently drop the cover slip in place. There should be no air bubbles trapped under the cover slip. To look at larger specimens, such as the Elodea leaf, place the leaf on the slide. Add a drop or two of water so that the sample is covered by water. Add the cover slip like before.
There you see the way that we would make a wet mount, either with the Elodea leaf on the right side there, or with some other aquatic features and stuff on the left. Here's what we're going to do in class or virtually for Cohort C students. For our wet mount preparation, you're going to make a slide with one small Elodea leaf, and you'll place the top of the leaf up on that slide. As it showed in there, we're going to take one drop of pond water from their mini habitats to make the wet mount. And as we do, we'll focus down through, find what we need to see, and complete part one of that Elodea worksheet. Here's our Looking at Elodea worksheet. It says, observe Elodea leaf layers. We're going to place the small Elodea leaf on the top upside, bottom side against the slide, and prepare a wet mount with pond water and a cover slip. You're going to first start off with 40, which is the lowest level, and then we're going to increase to 100. Then we will later increase it to 400. Use the fine focus knob at this point to carefully focus up and down through the layers of the leaf. Because it says layers, there's more than one layer. We'll be using this one. And then you need to describe what you see and how many layers you can see. Here's a visualization of what you might look at if you were to look at the Elodea leaf on the stage, on the slide, and then having your 10 power microscope objective lens as it's looking through. So on this one, the focal length will be 1 centimeter, so that's going to be how big it is from the end of the lens to the top of that particular leaf. That will focus on the top layer. Then you're going to move the fine focus so that the lens or the stage comes up closer together, and that will focus on the bottom layer of the leaf. So as you can see, there's only 2 layers of this leaf. It's unique in that way that it has a bottom and it has a top. Now, if you missed class or you're in cohort C, you're going to be using this virtual microscope, and that's at fosweb.com. Sorry, that's terrible handwriting on my part. You're going to go to the Diversity of Life unit, and you're going to find this virtual microscope. And when you do, you're going to be looking for the one that is the Elodea leaf or Elodea plant. And as a result, then it will show you, you can, the choose your subject is right here to choose the Elodea leaf. And you can move your objective lens or the stage. You can click on either one of those. And this is an illustration right here, is an illustration of the thickness of the 2 layers of the Elodea plant, the slide on the bottom and the cover slip on the top. To move up and down those, you're going to be using the coarse focus over here and the fine focus over here, and as a result, your image will change. So this is the image you're going to use to complete your worksheet part 1 and see what you see. Here's a closer look of what you're going to be seeing. This will look very similar to what we actually see in our class at 400X. There are boxes, what looks like boxes or bricks, like in a brick wall, in this Elodea leaf. You can see one right here. We can go around the outside of it, all this up in here. And another one, a brick-like structure. Not perfectly square, but you can see how they are boxed in and bricked in, etc. What are the boxes and leaves? They are our last important piece. They are cells. All living things have cells. And this Elodea leaf, are all these boxes are each individual cells for that plant. Here's another actual image. The previous one was just a computer-generated one, but this is an actual image, an actual picture of what some of those Elodea plant cells look like. And you can see the brick right here is a brick. Some of them are short, some of them are longer, some of them are here. They're all different shapes and sizes, but they all make that characteristic brick shape with them. So you'll use that observation to estimate the size of the cells and to draw them. Now we're ready for part 2 of the worksheet. Draw what you see in the field within its 400x. That is the most powerful. And you'll only need to draw 1 or 2 of those larger cells or boxes. And make sure that you estimate the size of one of the cells. Remember, we talked about field of view and that circle. That distance from one side to another changes as we change power. So in this one, the 400x, how distance across was only about 0.4 millimeters. So if that's all the way across the diameter, how much is just one of those bricks going to be? That becomes your estimate you're going to put on your worksheet. Here's that part 2 of the worksheet that you're working on. Describe what you observe in any of that. Remember, at high power, this distance of width or diameter all the way across is 0.4 millimeters. So a little bit less than a half of a millimeter is how big that item is. You're going to draw a few representative brick-like structures to scale. Whatever you see in this circle field of view, you need to draw it exactly. Make sure you use color and that you have good detail with it too. Next, question 7, how many of those bricks fit all the way across? How many? And then estimate the size of one of those bricks, just one of them, pick one. If the whole distance across is 0.4, how much is one of those bricks? And that is what you put in for number 8. Here's another illustration of what those Elodea leaf cell size might look like. The Elodea cells at 400x, you can see 1, 2, maybe 3, 4 bigger ones, smaller ones, all different kinds of sizes, but they all have the similar shape. So your task is to, let's say, take one of these right here. If you know this whole distance is 0.4, you can count how many there are and how big each Elodea cell is. Estimate. If I was looking, maybe approximately 0.1 millimeters. We'll go from there. So now that you've drawn them, seen pictures of them, looked at them through the microscope or the virtual microscope, are all the cells in the Elodea leaf the same size? I saw big ones, I saw small ones. So the answer with that would be no. There are different size ones. How many layers of cells are there in the Elodea leaf? Well, in that case, we looked at earlier. We said there were two layers, one on the top and one on the bottom. It's a very unique plant in that it's only 2 layers thick. Are the large cells on the top of the leaf or on the bottom of the leaf? Go back and look that again. Look in yours. And why do you think that is, that one would be where the larger cells are, on the top or on the bottom? And then again, how big are they? Usually we're looking at their length. I think we came up with that. It was about 0.1 millimeters. That's about how big an Elodea cell is. In the next lesson, we'll be looking at what specifically are those things inside the bricks and the boxes of the Elodea plant cells. As we go along, we'll learn some more. Thanks.